And it's great to see so many people here at Westmead Institute for Medical Research for our Science Week. My name is Diane Humphreys, and I'd like to start by acknowledging the Darug people, who are the traditional custodians of the lands and waterways here in Westmead. I also pay my respects to elders past, present and emerging, and celebrate their ongoing culture and connection to this land and water. I also welcome and acknowledge the Aboriginal and Torres Strait Islander people we have with us here today. I'd like to kick off with a short video about WIMR and what we do in Western Sydney and globally.
Greater Western Sydney's ascension to prominence in the Sydney Basin is underscored by a unique convergence of factors. With more than 160 different nationalities and the most extensive urban, Aboriginal and Torres Strait Islander population in Australia, it's home to 2.5 million people. It contributes to half the New South Wales economy, with notable infrastructure investments, including the forthcoming Western Sydney International Airport and improved transit networks set to further increase its significance and influence. As a result, it's not surprising that the Westmead Health Precinct is home to two of the largest teaching hospitals in the state, the CSIRO and Medical Research Institutes. But at its heart, and driving internationally recognised research, is the Westmead Institute for Medical Research, better known as WIMR. One of the largest medical research institutes in Australia, it has more than 400 staff and students across 11 centres. Closely affiliated with the oldest university in Australia, the University of Sydney, and Westmead Hospital, many of WIMR's scientists are also clinicians working in the Westmead Adult and Children's Hospitals, along with researchers across many disciplines. This means knowledge gained in the hospitals can be translated to the laboratory, offering a unique bench to bedside and back approach. This affords WIMR a translational advantage to precision medicine, forging breakthroughs in areas such as mental health, ovarian and breast cancer, heart failure, and antibiotic resistance, to name just a few. All of this, along with the diverse community it serves, makes the Westmead Institute for Medical Research one of the most innovative and groundbreaking research facilities in the world.
The structure we have here is that we've got 5 major research areas. Infection and immunity, so that includes HIV, COVID, multiple sclerosis and antibiotic resistance. We've got cancer, which on the 7th floor, the whole 7th floor here is devoted to cancer research. Liver and metabolic, that includes hepatitis, diabetes and obesity. Then we have neuroscience and vision, so that's brain function, mental health and vision disorders. And finally, cardiorespiratory, so that's sleep disorders that you'll hear about today, asthma, as well as heart damage. What we're all about at WIMR is precision medicine, and you may or may not have heard of that term. So traditionally, when patients are diagnosed, they were given a blanket treatment, a standard treatment that everyone got. At WIMR now and in the future, we're all about precision medicine. So that's taking the individual and their diagnosis, looking at their genetics, their environment and lifestyle, and then using data analysis to come up with personalised individual treatment. And that's far more effective and gain improved outcomes and also far less side effects. First talk today, we're very fortunate to have Adjunct Professor Ruby Lin, the leader with 25 years experience in life sciences, biotechnology and research. She's the genomic chair of the advisory committee here at Westmead, and she's also the deputy director of Phage Australia, and you're going to hear a lot more about Phage this morning. Ruby is a recognised expert in genomics, and has dedicated her career to scientific research, encouraging women into STEM, and she is also the inventor on several patents. Her presentation today is entitled Phage Therapy, so please join with me in welcoming Dr. Ruby Lin.
All right, so continue on what Diane said. I'm going to talk about how we use phage. Bacterial phage is a virus that kills only bacteria and doesn't seem to affect us humans or mammalian cells. So just to make it interesting, I am giving away phages, soft toys, for the best questions after this talk. So please pay attention. Okay, so what does this look like to you? It's not aliens landing on the moon. It's actually a phage sitting on top of the bacteria wall and trying to target and inject its DNA material into the bacteria. So as you're sitting there listening to me, you've got about 5 billion of phages on you and in your, especially around your gut microbiome. So phage has been around for a very long time. It's the most abundant entity on Earth. And it's co-evolved with bacteria probably for millions, maybe billions of years. Phage actually kills very quickly. They actually collaborate with antibiotics, and it enables them to work very well for bacterial infection. As you can see from this electron micrograph here, it kills the bacteria explosively, and it's able to amplify itself using the bacteria leftover material. So I'll come back to these 2 points later, but the main point for this slide is that it kills explosively, it amplifies itself, and which makes it really good when we're actually trying to make this into a medicine. So back in 2014, the head of WHO, Margaret Chan, actually foreshadowed antimicrobial resistance or antibiotic resistance. She actually stated that antimicrobial resistance would be the end of modern medicine, and as you probably know, antibiotics has been very useful in transplantation, in cancer therapy, in all of the surgeries that we use in modern medicine. But just to give you some stats, in 2019 alone, all of the antimicrobial resistant death is actually more than what we've observed for COVID. And of course, it will be in a few decades, less than 20 years, it may be the number one cause of death, exceeding cardiovascular diseases and cancer. And you can read about it in these papers I've listed here. But in fact, in India, we actually have patients that actually survived chemotherapy but died of infection. So that's how serious this antimicrobial resistant or AMR is. Okay, so back in 2016, the UK government actually commissioned a report, and Jim O'Neill actually reviewed this whole antimicrobial resistant landscape and pretty much have said that phage is actually a solution, number one solution to solve this. So how does it work? It's natural and it controls the bacterial population. It's very specific. So going on the theme of precision medicine, that what we would like to achieve at this precinct. And it's been used for over 100 years. So in countries like Georgia, Poland, Russia, for example, if you have a toothache, people will probably, the clinicians probably prescribe you antibiotics, right? In those countries, they just make you gargle phage. So you have it's a mouthwash, and that's it. So the cultural difference of use of phage has been around for over a century. And so how do we do it? It's pretty much knowing the right phage. So it's not just like, okay, you go to a nursing home, grab the sewage, extract the phage, and you go, oh yeah, I'm going to use this. So Kylie can probably tell you how we made the trip to the nursing home and grabbed the smelly sewage and got the phages out. So it's knowing the right phage to use in the shape of the biobank. So we actually have a collection of phages that would actually target most of the bacteria that we see, or most of the pathogens we see at the hospital. And of course, knowing the right targets. So we integrate with our national surveillance program. So for example, around 40 to 50 hospitals, if they have an outbreak, they send us the bacteria isolate to sequence, and we produce a antimicrobial resistant report for the government, and that forms part of our national surveillance program. So with these two knowledge in mind, we actually have a phage bank as well as a pathogen bank, and we actually grow the phages to a specific liters, which enables us to make a lot of dosages to treat the patients. And this is the generalized pipeline that we use. And of course, on top of this is the regulatory, the ethics, as well as all the integration between the clinical side as well as the research side, which I'll elaborate in this talk. So we have been using phages for probably John Iredale has treated the first patient back in 2007. I joined his group about 7 years ago. So we were able to secure a lot of grants that accelerated a lot of this. So I'll go through some of the high-profile cases that we have treated so far. So on here, we saw Josh when he was 22. In fact, John saw him 3 years before. He has infective endocarditis. So look away if you're a bit squeamish, but this is pretty much an infected aortic root and the valve that came out of Josh. And I've got permission to share this. So he was in and out of hospital, he was getting really sick, and we treated him with phages. He was able to 2 weeks of phages, and he was able to go back to work, and 3 years on, he's, healthy and he's playing rugby. This is Denvi. She was 7-year-old when she traveled back to India for a family holiday. She was involved in a car accident. They put a metal plate in for her on her left leg. She came back, but it was discovered that she had antimicrobial resistant Pseudomonas infection. The bone wasn't healing. She was on antibiotics for over a year. So can you imagine a 7-year-old with intravenous antibiotics for over a year? So by that time, the orthopedic surgeon pretty much said, okay, amputation, because the bone was not healing. So it just happens that Amane, my co-deputy, came to one of our talks and said, hey, Ruby, can we actually use phage for this little girl? So we did, and it was amazing that this expedited the legal ethics, everything to Ministry of Health, and we were able to get the phage into the country within 6 weeks and start treating her. So amazingly, we treated her for 2 weeks again, and now she's back to school and she's climbing like any 11-year-old would. This was taken about 2 years ago, so she's about 13 now. Rebecca, when we saw her here, she was 14. She has cystic fibrosis, and she's been in and out of hospital for 5 years. So just let it sink in. She's been at hospital, not going to school like you guys, since she was 9 years old. And she get hooked up to these machines for 5 years. So we treated her for almost a year with Phage Therapy, and here she is with her soccer coach. She can play Saturday soccer now, and she's well enough to go back to school and do, things like, any 14-year-old would. So I put up these cases to show you that phage is possible, and this is our latest one. Paul is a musician. He plays double bass in the Sydney Chamber Group. He also had post-operative infection, and this was a special case because we got the funding from New South Wales State Government to actually manufacture the phages ourselves here at Westmead Institute, and we're able to treat him for 4 weeks. And a year on, there's actually no signs of infection, and in fact, he's gone back to work. He's actually playing in a concert this Saturday at Springwood. So I mention these 4 cases because you can see that the treatment period ranged from 2 weeks to a year, and so it's very similar to how you would actually administer antibiotics. So we actually did some health economics. So for those of you who are interested in commerce and health, we looked at if we actually introduce phage therapy as a clinical service in public hospital in Australia, how much would it save for public hospital? And the stats are amazing. We would have avoided around 350 deaths and saved the hospital $67 million alone in that year, 2018 to 2019. And this is for severe acute infections. Okay, so for chronic infections like hip and joint surgeries and replacement, we would have saved around 1,900 operations, avoided around 300 premature deaths, and a staggering 123 million saved if we actually just introduce phage therapy as a clinical service in public hospital in that 1 year alone. Okay, so from what you've heard so far, I have convinced you that AMR or antibiotic resistance is a problem and that phage therapy is a solution. And so to that end, what I've been building with John Iredale is an integrated network from engaging with the government and our regulatory body, TGA, as well as the researchers with the research institute, universities, and getting people interested in this type of technology through industry participation. So as you can probably appreciate that, when you actually picked a phage from the soil or from, your compost or from sewage, of course you can't actually use that. There would be a lot of molecular tools that we would need to use to bring it up to a point that it is safe to put in humans, so intravenously, etc. I'll show you in a slide in a minute. You don't have to be able to read this slide, but this is just to show you that we've got a lot of stakeholders involved. So it's not just a 2-people show. It involves a lot of people with the buy-ins. And what we're trying to achieve is pretty much, instead of addressing individual cases like the ones that I've shown you in the previous slide, trying to bring that into the mainstream so we can actually democratize access. Okay, so this is the buzzword from bench to bedside, but we truly are doing that here at Westmead. We actually, every phages that we have produced so far in the lab, we have made it to the point that it is safe to put in humans. And so it ranges from phage hunters like this, like us. So this is us in the basement of the hospital below the dental school collecting sewage. And I can tell you that smell lingers. And I can still remember that smell. And this is where, so you might ask, why do you need to go there? The phages that we picked up from that kind of sewage is pretty much the phages that will actually target broader range of bacteria. So we go from this to biobank. So ideally what we like in the future is have a selection of phages in our arsenal and actually deploy them. If we actually say, hey, there's an outbreak in E. coli or there's an outbreak in Pseudomonas, we will actually have a range of phages to actually deploy straight away. So from there, we actually do a lot of lab, and of course we are involved in a lot of education in terms of trying to bring people to be aware of what antimicrobial resistance is, what phage is. And this is Karen, our clinical coordinator, using a Oculus. You guys, the school kids, will probably be aware. So what we've done is we actually have made an education package, and where it's an immersive experience that enable adults and kids, patients, to see what phage therapy or experience what phage therapy is going to be like as soon as they step into a consultation room and as well as the hospital. So from there, we have here Dr. Stephanie Lynch. She's actually in charge of all of our phage susceptibility testing and, of course, phage production from Rebea. And we do a lot of genomic sequencing. So when I actually mentioned, or when Diane mentioned, precision medicine, molecular tools, we do a lot of genome characterization. And it's to the point that you can actually identify a lot of antimicrobial resistant genes, as well as toxins or any other genes that it might cause issues if you actually put the phage into the patient. So all of the stuff that we're doing and using modern technology is try to make sure that the phage product we put into the patient is safe. And of course, we do a lot of R&D, research and development. So how do we do this? We actually have a very permissive TGA, by the way, so I'm very appreciative of them. They were very open to advanced therapeutics such as phage therapy, and we actually work with them on how to regulate phage therapy. So I actually didn't mention earlier, but a lot of these advanced therapeutics or experimental therapeutics are actually not regulated. Specifically phage, they are different regulatory kind of models around the world. For example, Belgium Magistral model compared to us and FDA. FDA have an expanded access scheme, so pretty much all the orphan drugs and all the experimental drugs go through that pathway. And why do we need to do this? Every medicine you have taken have gone through a very stringent pathway of regulation to make sure that it is safe for you. So we are doing the same thing for phage. So in this case, we actually have devised a standardized monitoring protocol that we can actually carry it out. And the beauty of this protocol is that it enabled us to actually collect a lot of data. So everything that comes out of the patient, every fluid, we collect and we do clinical research on them to make sure that the phage is actually safe for human consumption. So because of this, and it's under the clinical trial framework, we're able to build trust with the prescribers, the clinicians, and also the community. We actually have a lot of adoption from cystic fibrosis community because they are all early adopters. They like all the new technologies. And of course, build trust with the regulators. With the clinical outcomes, we're able to actually address the safety as well as the efficacy of phage therapy. And so to that end, we actually have a lot of buy-ins from clinicians and hospitals. We're able to see all these major cities that actually have adopted or they have site-specific approval to carry out phage therapy or STEMP protocol. And the way we've done it is very different to how other people would have carried out this kind of experimental therapy. So we actually have got all these countries, for example, Israel, US, Korea, Denmark, Canada, UK, Singapore, and India. They actually all want to adopt this STEMP protocol and how we've integrated and being inclusive of everyone in this journey. So I can share these slides. I've put up some references in this slide, but it's just to say that Phage Australia was mentioned about 22 times when the UK Parliament did an inquiry last year on the antimicrobial resistant potential of phage. So we were heavily featured in an international platform about how we actually approach this in Australia. So I'm very proud of that. Okay, so if we say, hey, phage is great, we actually have to convince the prescribers, right? So what we've done is we carried out 2 surveys for infectious diseases clinicians and asked them how do they feel about phage therapy and how much do they know. It's not just trained clinical microbiologists that know about pathogens. So what they have come back with is that they would like to try phage therapy. They know about the safety of the phage therapy, and there is a unmet needs for all these diseases. Specifically, and you probably wonder why I mentioned prosthetic joint infections when we did the health economics. And one of the reasons is that they can see that those cases, the prosthetic joint and devices infection are the ones that actually are causing the burden on our public health system. Okay, so this is a little bit of nerdy stuff about how we actually build our collections. So we would actually have a clinical triage, and then when it comes back, so the clinician would decide, okay, this patient needs this patient have bacterial infection, antimicrobial resistant infection, and they've had optimal care, but the antibiotics is not working. So then they will actually contact us and say, hey, is this patient, can they actually go into phage therapy? And so there is a clinical screening procedure, and when the patient isolate comes, we do a phage screening. So in that case, it's like, for example, if you actually have a Pseudomonas isolate, we will screen against all of our Pseudomonas phage collection. So as you probably can appreciate, that would take probably a week or a few days. And of course, when we pick the right one, so you would do a plug assay. So traditionally, you probably have seen photos of people holding an agar plate with a plug there. So those plug are the ones how we actually determine if a phage is actually killing that bacteria, the tick. So once we've identified a phage, 1 or 2, then it goes into phage production. And then, of course, we do very stringent QC, so quality control. And all of this runs in parallel. So the clinical cases actually run in parallel with the biobanking pipeline. So in this case, we will actually bank the patient isolate because those bacteria are actually very important for us to do research. And as part of the National Surveillance Program, it's actually important to say, hey, back in 1980s, we have, say, for example, 200 Pseudomonas antimicrobial resistant isolates, and that would be our reference. So then if we actually look back and compare to 2024, we would say, hey, we only have maybe 150 AMR Pseudomonas isolate, and then you can actually do a lot of retrospective and a lot of tracking of how they have evolved from back in 1980 to modern days, to date. So what I mentioned about biobanking, what we have like to do is have a panel of all of the phages. At the moment, we've got around 3,000 phages in our collection, and we've got around 50 phages that are therapeutic. So we're able to, if the clinicians tell us, okay, this patient have a specific infection, and if we have phages, we can actually deploy that within 2 days. So in the olden days, the cases that I've showed you, it took 6 weeks. Some of them took almost a year to get the right phage to treat the patient. So I hope this slide actually, the message is actually, if we actually have the collections, we're able to react very quickly or respond to the cases very quickly. This is the physical phage biobank, so you can probably appreciate that you need to label things systematically if you actually have thousands and thousands of samples. So this is just to show you that it's very elaborate, and we have to do it systematically. Okay, so precision medicine, of course, you need molecular tools, and one of them is next generation sequencing. I hope all of you will have come across this in the media. So we heavily rely on genomic sequencing in terms of sequence it to a depth that you can identify gene-to-gene interaction. So in this case, we have a pipeline that can identify lysogeny markers. So remember when I mentioned that the phage can kill bacteria very quickly and explosively? So that phage would have been classified as lytic. When it's lysogenic, it means it can actually integrate itself into the bacteria. So you don't want that, because if you imagine I give you a phage and it integrates into the bacteria that you already have in your system, that's not a good news. So genomic sequencing enables us to pick out all these markers within the phage itself. We can pick out virulence genes, antimicrobial resistant genes, and any other stuff that we deem not safe for the patients. And this is the assembly. So it's quite involved, but we've got domain expertise in this pipeline. This is just to show you how we directly translate it, but I actually put this up to show you that it's not just you produce the phage and go, oh, okay, off you go, you can put it in patients. We actually produce 4 reports, and these 4 reports is actually presented to the drug committee at the hospital and with the ethics committee every time we actually treat a patient. So the stringency of it is pretty much to make sure that the batch that we produce is safe for the patients. Okay, so these are some of the stats. So far, because of the funding we got from the state government as well as philanthropic funding, we were able to expedite locally produced phages. So we actually produce them here at Westmead Institute. So far, we've produced 44 batches, and we've got produced 22 phages, single, so it's 1 phage for 1 bacteria. Or we can make cocktail. So it sounds cocktail. What is it? It's a dose that contains more than 1 phage. So it can be 2 or 3. In our cases, we actually have made a cocktail of 4 phages, for example. Why do we need to do that? It's actually increase the range of how the phage would actually attack the bacteria. So far, and it's very versatile in terms of as a drug, because you can formulate it in a very different ways. So you can have intravenous, which is the main ones that I told you about, so directly into the bloodstream, or it can be instillation. So we've made phages that can go straight onto devices that are attached to the patient. You can take it orally, or you can nebulize. So for a lot of people with lung infection and cystic fibrosis patients, we actually nebulize the phages. Topically, you can make a cream if you have ulcers or infections on the skin. It can be topical and of course combined. Okay, so how do we make this sustainable? Pretty much the pathways that we are exploring is, of course, the integration, which set us apart from all the other operations around the world. We've got funding from philanthropy and, of course, with you guys coming here, community involvement, because we want to make sure that you understand antibiotic resistance is an issue and that, phage therapy is a solution. A lot of times we trying to make investment opportunities. Okay, this sounds a bit corny, but to make all these happen, it costs a lot of funding to make this happen. So when I mention about, investment opportunities, have people that are interested in part of the operations to come in and help us expedite operations. So, for example, we have Illumina that donated their sequencing for us to accelerate sequencing. And of course, that leads to creating different income and business case, and you probably can read about this in a lot of our white papers there. For me, I like to create a lot of partnerships. So specifically for the school kids here, we are actually kicking off a bioprospecting program. So that enables you to, we'll send you a kit that enables you to go and collect, whether it's from the river or from your backyard, your compost, send the samples back to us, and we're able to extract the phages from those samples, and you get to name them. So part of that is getting community involvement, right? But it's also to increase our collection of phages. So part of this is citizen science approach. So for the science nerds, harmonized SOP, what does that mean? So we might have a specific way of doing our experimental design. So we have a specific way to do, say, phage susceptibility testing. Ideally, we would like to harmonize it around the labs that are involved in phage therapy, so we can actually, introduce standards, and we could actually exchange phages directly. And of course, part of this talk is that workforce training and education. So I think I will stop here. This is my acknowledgement slides. As you can see, we've got a lot of people that are involved in this operation, and of course, all the support we're getting from the Westmead Institute itself, as well as our state government, of course, our federal government, and all the scientists and industry as well. Thank you for your attention.
Thank you so much, Ruby. I'm sure we'll be hearing a lot more about phages into the future. And we've got about 15 minutes now for questions. We'll bring the clock over mic around the brief so that everyone can hear. So does anyone want to go?
Yeah.
Are there any side effects?
They are side effects only based on the purity of the phage. So sometimes it will cause the fever, and we've seen that, but it's a natural immune response, and we've observed it in adults and kids as well. In fact, I was able to identify quite a few gene signatures associated with innate immune response, as well as adaptive immune response. I got a patent out of it, so it's not bad. But to your point, it's a very good question, because a lot of people say, okay, it's safe. And, I've shown you in the slide, you can use it for 2 weeks, or you can use it up to a year. So it's like antibiotics. And with anything, it would definitely develop some sort of resistance. But in this case, we follow the patient all the way to 90 days. And some of them you see, 3 or 5 years, they're all doing well. So I can confidently say that it's safe.
So what do you think is the biggest prohibitor limiting phage therapy from entry into the clinical setting, seeing all of this positive research? I know you mentioned cost and the FDA access scheme as some prohibitive factors, but which one do you think is the biggest component, which is...
Very good question. I think it's adoption. So at the moment, we're talking to quite a few regulatory bodies around the world. So, for example, our collaborators in Israel, they've convinced their government to actually participate in phage therapy, and they're actually implementing it into their hospital. US is a bit funny thing. They don't have universal care like we do. So very different approach and different scheme to go through. But the economics is the main one. So there are a few phage companies out there, but they have adopted a very pharma model. So as you probably can, see from my slides, phage is a life product, so they don't know how to deal with that. And phage is auto-dosing. So if you have one dose, say 10 to the 9, it can amplify itself. So when we actually treat the patient, we would treat the patient and maybe 2 or 3 weeks later, we can still find the phage in the patient. So it's auto-dosing. And so a lot of, PK/PD kinetics and pharmacokinetics needs to be done. And so when it's like that, the regulatory body find it really difficult to regulate. And so that's why there's different schemes. The Belgian magistral model is quite good. So they pretty much are saying, as they don't care where you got the phage from, as long as the phage that you put in patient is clean. So by clean, it means, you have to go through a good manufacturing process, but they don't certify it as a GMP product. They certify it as it's active against that bacterial infection. So that kind of regulation is actually very good because the point that you pointed out is economy of scale. At the moment, we're treating individual cases, so it's very bespoke. I know it's precision medicine, but it's very expensive. If you actually grow things, like with anything else, if you can grow in big batches, of course, it is cheaper. So ideally, we would like to get to the point that it's around the same price as antibiotics, so that it can, massively implement it into public domain.
Thank you.
Yep.
You said they had about 3,000 types of phages, so that different phages can target different types of bacteria.
Yep.
How do they target different types of bacteria?
We don't use, say, for example, Pseudomonas phages for E. coli bacteria. So you will actually have to have a specific phages against a specific bacteria isolate. So, for example, you will have E. coli. I'm no microbiologist, but if you actually have E. coli, they belong to different clades, and you will have specific phage that will only kill, say, clade 1 or clade 2. So as you can appreciate, because they have co-evolved, every bacteria will have a specific phage that will actually target it. So when I say 3,000, other people might have 50,000 phages in their collection, and they would target different bacteria.
Is it likely in the future that the phage type therapy could be used for other than bacterial infections, other conditions that people have?
It's a virus that targets specifically for bacterial infection, so it will only target bacterial infection.
Not cancer or anything like that?
No, but if the patient is actually under chemo, that we have quite a few cases like that, and their immune system is down, they're prone to infection, then of course phage can be used to protect the patient from the bacterial infection. So it's a good point that I implied it, but I didn't emphasize that in my talk, that you can actually use phage as a prophylaxis. So you can actually treat a hospital or treat a sink that's more than likely to carry bacteria. So you can treat it prophylaxis way. Yeah.
Yep.
You mentioned previously, and I don't know if it might be answered in the other adoption question, that in other countries in other countries they use phage therapy through like gargling phages. What are the cultural differences that kind of reduced our adoption of it, or are there any stigmas around using phage therapy here?
Well, I don't want to speak out of turn, but when Fleming invented antibiotics, I think it was back in the 1930s, like World War II, and of course Stalin endorsed phage therapy, and the Western world just decided, okay, we're just going to use antibiotics. And I think that's what happened, not sure, and I wouldn't make a public statement about that kind of stuff. But the adoption So I've been to Georgia, Tbilisi, and I actually visited Iliava Institute because they've been using phage, and in fact it was their 100th year anniversary last year. And it's amazing. They've got a whole collection of phages there, and they've got, very rich history. It's all in Russian, of course. And they, for every ailment they've got, so if they've got an ulcer, if they got, a graze, they don't actually go straight and reach out for antibiotics. They actually reach out for phage. So I was given, a couple of vials because, when you travel, your stomach is not so settling. The director of Georgia Iliava Institute gave me a vial and go, just take this, Ruby. And I didn't, but in hindsight, I should have, just to see, what it's like. So the cultural difference does affect adoption, but I think because we're trying to bring all the modern, utilizing all the modern technology to say, hey, look, we've sequenced it, we've, done all these experiments to show people that, it is safe to use. Hopefully there will be more adoption for it. And then going back to your question about, applying it. So this talk is heavily on, clinical service in human health, but phage has actually been used to clean oil pipes because it's still, bacterial biofilm. It's been used in aquaculture. It's been used in all the other industries. So, for example, dairy cows that get mastitis, it's a bacterial infection, like in Streptococcus uberus, right? So you can actually have a phage that treat that condition before they pump, the animals full of antibiotics. And as you probably can appreciate, that goes into our food chain. So, it accumulates in us. So a lot of animal health people and a lot of industry people are aware of this type of application. Yep.
Does phages mutate?
Yes, they do. Like anything else, they mutate. So bacteria and phage, they co-evolve. So of course, they would actually develop ways to avoid each other's arsenal. And by doing sequencing, we're able to pick up mutations and anything that they would change in a way. And they can actually co-evolve very fast. So one of the branch of R&D that I haven't mentioned in my talk is that you can actually do forced evolution. So you can actually force evolution in the lab in a way that you can broaden how a phage can kill a selection of bacteria. So you can actually broaden and force the phage to evolve in a way it will kill a lot more bacteria than it would originally have. So very good question.
I just wanted to know, and with hip and knee replacements and infection, and your just been deployed there.
Yeah, we have. So in the 42 patients we have treated so far, we had quite a few patients that had chronic infection with knee and joint replacement. So I think one of the slides I showed direct installation with actually just pour phage directly onto the infected area. So and it's been successful. Yep.
Are there like specific circumstances in individuals that they reduce the effectiveness of phages on them?
Yeah, so good question. So you're talking about anti-phage antibody. So we actually have got another branch of R&D that looks at it. So with anything that enters your, foreign body that enter your system, of course your immune system is going to respond and it will actually form antibodies against it. We actually have detected anti-phage antibody, but it's so low that it is still safe to use phage in the patient. So remember right at the beginning of the talk I said, as you sit there, you've got 5 billion of them on you and in you. So a lot of phages exist in us as prophages. It's integrated into the bacteria genome. When you get stressed, it comes out. And then when you're okay, it goes back into the bacterial genome. So you will already have a very low dose of immunity against it. So there is that, like kind of minimum level of anti-phage antibodies in your system already. But it's not to the point it's detrimental to you. So for example, with sepsis, the patient actually don't die of the infection. It actually die of overstimulated immune response, what we call it in cytokine storm, that actually causes organ failure. So it's not to that severity, but you do have that low level. Very good question. Yep. Oh, hi.
How would you instruct the phages from
Oh, okay, yeah. Nerdy question. Very good. Okay, so you will get the water, and then you will actually filter using 0.22 micron. So you use gravity, and you will actually filter the solution through, and from there you can actually find phages. But specifically what we do is you would have, plated out agar plates. You know what agar plates is, right? So yeah, it's a petri dish with, yummy agar in there. You actually put a lawn of bacteria on there. So because we don't know what phage you're going to get, so you actually put a mix, say you put E. coli, you put Pseudomonas, you put Klebsiella, and then you drop that solution that you have filtered on there. So we call it a spot test. And then when you get a plug, it means the phage is actually eating the bacteria. And then with that, we pick them out, sequence them, and then we identify the phage. That's how we do it.
So there's so many intermediate steps in the development. Do you see some of this stuff becoming automated or fast-tracked at some point?
Very good question. So remember how I said sometimes it will take 6 months, sometimes 2 days. The one that's 2 days, of course, we can expedite it because we already got phages in our biobank. The ones that we don't, we would actually need to go out and extract them and do all that. And definitely, I'm all for automation, right? At the moment, for example, it would take Steph probably 2 or 3 days to do the spot test and then do the kinetics, and she would actually, 96 well plate, prepared it like this. If it's automated, of course, it's going to save a lot of time, but that's what we're trying to do at the moment. Very good question.
Those combinations are getting smaller, so you're I would almost imagine this building into a library where you're seeing patterns and then going into record somewhere that eventually streamlines some of your processes.
Yeah, very good question. So the physical side, yes, you will streamline it in a way. So I had to give you the example. It's at McDonald's, right? You have a catalog. You go, I'll have that, and that. So ideally, you want to get to the point that with the biobank, I'll go, okay, I have phage 1, phage 5, and phage 13. Put it all together and give it to patient number 3. I will have phage 60, phage 57, mix them all together to give it to patient number 1. That kind of approach. We're getting there. But you actually highlighted something I didn't say, and I think the school kids need to be aware of. So with all the stuff, all the data we collected, especially genomic data, we collect a lot of data. And part of it, there's a trend going towards prediction, machine learning. So, you hear about AI, the buzzword and all that stuff. We actually use a lot of artificial intelligence in a way that can cleverly predict. So to your point, in future and probably soon, we don't actually have to go into the lab and do the old school spot test. We can actually say, okay, we've got the genomic data for the phage, we've got the genomic data for the bacteria. We can predict how they're going to interact together. And then it will actually accelerate a lot of processes that we can select phages for therapy. Yep.
Based on that, thank you. Based on that previous question, do you predict that in the future there'll be institutions specifically for keeping phages, as in a library where you can take out specific phages out of the potentially thousands?
Actually there are companies now that goes around the world and say, oh, you've got phages in your freezer to, researchers, and then go, I'll buy them from you. So there's a company, it's recently got brought out by Biomex, APT, Adaptive Phage Therapeutics. It went around countries and actually pretty much said, okay, I'll buy your phages to add to our collection. Is that what you mean? Yeah. So a lot of researchers actually do have a selection of phages in their freezer somewhere. So part of the networking, the network that we have established is try to have involvement from all these researchers, because it is actually quite costly to go out and do all the bioprospecting. It's very good exercise for the kids that want to be involved in science, but the effort involved in characterizing it all the way to a clinical grade phage is, you probably can appreciate it's very expensive. But if the researchers in different labs around the world already have it in their freezer somewhere, it's actually easier for us to reach out and actually get them. And I must say the phage therapy community is very inclusive, and, they're all very collaborative as well. So we all talk to each other.
They've just got time for one more question.
I will be sticking around for morning tea, so if you want to come up and ask questions. I think these guys are competing for the cell toys.
It's not so tough. Who's the ones to love for?
You took it up.
Thank you for your presentation. It was so informative and fascinating. I'm a teacher, and I have had a number of students here today, but just on behalf of all the high school students here, I know that some of them will be thinking, wow, this is amazing. Could you speak a little bit about how you've got in? What was your career pathway into what you're doing now?
Oh, wow. How long have you got? Well, I did the standard high school undergraduate degree and got a scholarship to do a PhD, but at the time I was like, don't know what to do. So please don't feel that by the time you're 17, 18, you actually know what you want to do. I think a lot of careers, I gave this exact talk at Hills Grammar probably a few months ago. I think you need to experience it. So, Westmead have got a very good program for students coming in to do some of the internship. So you can come in for 3 or 5 days just to see if you like this kind of work. For me, so after PhD, I did postdoc and, the standard research career track, and I was able to get funding. But then, I felt like I was a bit bored, so I moved to a different area. So initially, I was actually trained in genetics, in genomics for cardiovascular diseases, and I moved into running a clinical trial for asbestos diseases. And then so I was able to pick up all the different skill set along the way. And when John actually, I got this job from LinkedIn, so use your LinkedIn. So I contacted John and said, look, you're looking for someone that can run clinical trials and know about biobanking and have got all these molecular genomics background. That's me. So I kind of accidentally fell into this job. And I must say, all the jobs that I found so far is by serendipity. It's always someone I know that needed my skill set. And that's not to say that all of the stuff that you're doing at school at the moment, it will come in you. So, you learn how to write exams, right? So you've got 90 minutes, you know how to manage the time. And if you actually do biology and chemistry, you might not end up doing science. You might end up doing a creative degree, but, it's also useful because the skill set you've learned is transferable. So my career advice is that do something that you enjoy. But even if you don't get the subjects you wanted at university, say if you want to go to university, it's okay because it's transferable, that all the stuff is transferable. I think they did. Are you guys millennial or alpha? Like, which generation are you guys?
Z.
Z, Gen Z? Okay, so Gen Z, I think they did a survey that you probably, well, your first job is not your final job. It's not like, my parents, they stick with one job all throughout their life. You probably have 5 to 10 different jobs. So, it's not end of the world if you don't get the subject you wanted at university. For those guys that doesn't want to do it, for example, in New South Wales, there's actually a program for training workforce, for specialised workforce. So it's not, you have to go to university. They are jobs that going into manufacturing, for example. So I'm part of the New South Wales government's roundtable GMP, good manufacturing process discussion. And part of it is trying to get the students to go through from high school to, university or to other training to help with the industry. So it doesn't have to be a tertiary degree. It can be, a work-specific trained certificate to help. So keep an eye out for those type of stuff because, it's not everyone can get into med school, right? Yeah.
Thank you, Ruby. Well done. You had a fantastic presentation on Phage, that a great career advice. So for most of you that want to pick Ruby's brain, please hang around. Please join with me in thanking Ruby for a fantastic scientific presentation. Thank you.